It's Monday morning, also for me, so what I would like to do is to tell you a little bit about tumor biology in the first hour of the lecture, and in the second part I would like to dive into more examples why I desperately need bioinformaticians like you. So I'm very happy that the Master Bioinformatics exists, that it's growing every year. And if I would have been a student today, I probably would have taken the bioinformatics courses that you take today. But now I would like to explain to you a little bit about oncology. And who has a little bit of background in oncology? Some? Yeah, I see a few hands. Yeah, so I know some of you have had some more biology past, and some others will have some more computational past. Is that correct?
Yes.
So just would like to explain a little bit what tumor biology is about. And so actually, to start with, this is a nice Monday morning picture, and the question here is on the left. What is it that you see? I know it's Monday morning. Just give some answer. You see? You see kind of the origin of life, isn't it? This is how we all started. And then on the right, this is our Monday morning, how we are sitting here today, right? And the interesting thing is, something happened in between. And the question is, what happened in between this origin of life and this Monday morning? What happened to that cell, actually?
Started dividing.
Yes, dividing. Proliferation, yeah. That's good. So then you have a big clump of cells. That's the first thing we can think of Monday morning. One cell becomes many cells. Okay, here we have the big clump of cells. We're not a big clump of cells. What happened? What else? Differentiation, exactly. So there's all kinds of changes to these cells that prevent us from being a clump of cells. What else? Okay, you discuss what comes up next. Some cells need to die off once in a while.
Yeah.
And then, there's like communication between cells and things,
Mm-hmm.
But actually the question mark is why it's interesting to study cancer. Because when you study cancer, you basically study what goes wrong. And so you learn actually what goes right. So this is basically the summary of the first lecture. But let's get into a little bit more details. So this little sperm cell brings basically in DNA. And this oocyte also has this DNA. So apparently you have a copy of DNA from your father, from your mother. And everything that you need is in that strands of DNA, right? Isn't that amazing? I think that's amazing. But that's how it is. And then of course, you need to translate this DNA into RNA, and then the RNA is going to be translated into proteins, and these proteins are a little bit functional units, and you put them together and you build cells, and these cells build up us, right? That's kind of the summary. So, when you think about this clump of cells, it is not necessarily a clump of cells, but if we are these adults, we exist of this number of cells that somehow differentiate, that somehow die off when it's needed, that somehow proliferate, and then there's some stem cells that make sure that whenever needed, there's going to be some new cells there. But somehow this is all in balance. I yesterday had a reunion of my classmates of the basic school, elementary school, we all turned 50. So I must say some things change a little bit, but really the majority doesn't. I guess my body, all my cells have been replaced, my molecules have been replaced, but basically we're discussing homeostasis. Basically we are capable of maintaining ourselves. Now, when people think about cancer, they sometimes say cancer is a disease of the genes. I think this is now old-fashioned. I think we should now say cancer is a disease of the DNA. Because I think there's more in DNA than just genes. But why do people say that? Why is cancer a disease of the DNA? I mean, let's face it, these proteins are the working guys, right? So if you break up these proteins, doesn't that really matter? So why don't we say cancer is a disease of the proteins? Why is it the disease of the DNA?
Proteins are derived from the DNA.
Sure.
Actually, proteins are.
Sure, so you can do something to the DNA and then the protein changes. That matters, right?
Mm-hmm.
So why isn't it a disease of the protein then?
Because it's not the protein's fault.
Well, if the
Protein is not the origin of the disease, it's the DNA mutation.
So, if we take this oncogene and we mutate it, this is what goes wrong in the cell. So why is it not the disease of the proteins in the way we talk about cancer?
You can't cure the protein.
Sorry.
You can't cure the protein. You can.
Well, people try. People try.
They try.
Yeah, many drugs connected to these proteins. Yeah. But the tricky thing is, what if I break a protein? What will happen to the next cell, the progeny cell?
They'll still produce the protein.
Yeah, but if I break the protein, will the next cell have, when new protein is generated, will this be a mistake and a fault? It's the defective protein. What happens if you alter the DNA? And you think about the progeny cells.
All subsequent cells will have those alterations.
Exactly. Yeah. So when we discuss cancer as a disease of the genes or of the DNA, then really the key that what we are referring to is when you change something in the DNA, it will be anchored and it will be transmitted to the daughter cells. And of course from there you will have changes in the RNA, in the proteins, in the cells, whatever functions that follow. Yeah. And what's the consequence? The consequence in case of cancer is somehow you are losing mechanisms that make sure that homeostasis takes place. So homeostasis is kind of lost. There's a disbalance in what happens to these cells. Do we get cancer from a single DNA mistake? No. Are unlikely. So, one thing that I want to discuss with you next is that cancer is a multi-step process. I happen to work in a department of pathology, so in my surroundings, many pathologists who like to look through a microscope, look at sections that are stained with H&E, and you get these pinkish stainings. This is a normal colon with lots of mucus. Those are the white blobs in these cells, very differentiated cells. When the differentiation gets lost in these benign lesions, adenomas, you can see that there's a higher cell density, but then these become invasive, become cancers, and then it goes all wrong. And so, in the 90s, there was like a first model of steps in which the DNA can be altered that could turn a normal colon into a colon cancer. And this is like this first multi-step model. So someone, like Vogelstein, proposed you will have some changes in genes like the APC tumor suppressor gene, and you may have a mutation in KRAS and a mutation in p53, and then it turns from normal to benign to malignant lesions. And so I think this model has changed, but not conceptually. I think by now there's many more genes that are identified as being relevant to tumor development. But obviously there's multiple genes that need to be affected before cancer can arise. So I'll make use of some cartoons. These is these mucus-filled differentiated cells, and these are cancer cells with their ugly nuclei and lack of differentiation, right? Lots of DNA here. And so what I want to discuss with you next is that we often say that cancer is a complex problem. Now, what I want you to learn from this lecture is, what do we mean with complex? When it's Monday morning, complex just means I haven't got a clue, I don't want to think about it, get lost, it's complex, like get away. But complex can also be, it's complex and I actually understand why it is complex. And this is what I want to explain to you, so that after this hour you have a perception why this truly is complex. So here we have this normal differentiated cell and this cancer cell, and then we wonder all kinds of biological processes that have changed. And there is these things that you already mentioned, there's going to be more proliferation, lack of programmed cell death, apoptosis, things go wrong in the cell cycle, telomeres are changing, telomerase is actually being upregulated. There's this extracellular environment where things are changing. Blood vessels are invited to come in, angiogenesis. There's rebuilding of the extracellular matrix. Cells start to move, start to invade, start to travel. So they start to metastasize basically, and on top of that, they fool the immune system. That's a lot of biological tricks that kind of take place in this transition from a normal cell to a cancer cell. And so in 2000, there was a review by, let's say, two of the most famous cancer scientists, Doug Hanahan and Bob Weinberg. And so they wrote this review. It's like a mini book about tumor biology. And they came up with this wheel of processes that are altered. But developments go quick. So in 2011, they wrote the same review, but they updated it. For 10 years, they had pissed off completely the immunologists because, avoiding immune destruction was not part of the original wheel. And some people working on inflammation and metabolism, and they even forgot about genome instability. But nevertheless, if you would like to have like this mini class on tumor biology, I would advise to read the review from 2011, because really it's nicely organized, all these different biological processes that are altered, and at least for the year 2011 reasonably updated. You will see this wheel of biology returning in many presentations on oncology. So it's a review that is very frequently used. Now when you think about these biological processes, these are not regulated by a single gene. So you get all these beautiful maps, and you can find them in many flavors, and I hope some of you are building them in a sensible way to think about how can I combine genes or elements in the DNA to bring together functionally a lot of these units in the DNA that work together. So, there's lots to say about just apoptosis or just proliferation or anything that has to do with angiogenesis, right? But if you think about so many factors involved in each of these biological processes, it also means there's a lot of anchor points where things can go wrong. So when you have DNA mutations in patient number 1, tumor number 1, you may have some changes affecting proliferation, apoptosis, cell cycle, telomerase, stroma, etc., etc. But now if you take tumor 2, patient number 2, you may see that the same biological processes are affected, but in fact a completely different combination of genes involved in each of these processes may be damaged, right? So here you can have 2 colon cancers. Superficially they look the same. When you dive into the genes that are altered, They may look completely different. And this has consequences, right? Because if you think about prognosis for the patient, if you think about treatment of the patient, of course it matters how exactly this particular tumor has been affected by these DNA alterations. And then it gets worse, because you can think about how to molest the gene, and there's many flavors in which you can do that. This can be point mutations, this can be copy number gains and losses, amplifications, methylation, post-transcriptional modification, germline variation. So there's many ways in which you have to look at each of these genes, DNA elements, in how they actually may cause or may contribute to the tumor biology. Clear?
Yes.
So here we look at the cell, and don't read all these names. I would say this is like something how we wire up a telephone network or something like that. So you see all these cables and connectivities inside, and then in patient 1 this could be the disconnected cables, and in patient 2 it could be like this, right? Summary of the previous slides. So let us go through each of these biological processes as mentioned in a little bit more detail. Proliferation is the first one that came up this morning, and that's often the case when we discuss cancer, and of course for good reasons. So here, if we look at a cell, and one day something goes wrong. There's this mutation in this cell. So now let's say it's a single tumor cell. And then what happens is that the single tumor cell starts to proliferate. And let's say that when there's 30 doublings, we have 10 to the 9th cells, about 1 gram of tissue. And if you think about 1 gram of tissue, this is something you may be able to see on a CT scan or on an MRI. So let's say a ½ centimeter to a centimeter lesion is something that the clinician would say, this is early disease, small tumor. But as a molecular biologist, I would say, well, I don't know. We have had here 30 doublings to get a gram of tumor tissue. Now if we would move on to another 10 doublings, this would be 1,000-fold more cells, 10 to the 12th. Would be a kilogram of tissue. That sounds like a lot, huh? And so for the clinicians this would be a huge tumor mass, but for the molecular biologist it feels like the majority of molecular events may have taken place in these first 30 doublings. Because now if we look at a single tumor cell, you can have this clonal expansion, but then in some of these cells new mutations occur, and then you get these subclones, where you get subclones, and you get this heterogeneous blob of cells in which multiple alterations take place. And you as bioinformaticians can make beautiful plots like these. I turn it a little bit, but it's great to have this kind of visualizations. I love it the way that in your work you have to think about how to visualize what we see in terms of the data that we analyze. But this is like looking at this clonal expansion, existing subclones over here. You give some therapy and then some of these subclones grow out, others don't, etc. Nice way of looking at it, but basically the same process as kind of described here, at least this part. When something is unclear, you raise your voice, right? What else about proliferation? Well, there is this cell cycle that cells need to go through, but then there is signals from outside the cell. And some of these signals are growth inhibiting, like TGF-beta. Others are growth enhancing, like EGF. So basically cells listen to signals that come from outside the cells. So there can be pro-proliferation, there can be anti-proliferating signals. And as long as these cells listen, that's pretty fine. How do these signals come about? Well, you have sometimes autocrine signaling, so that cells actually stimulate themselves. Paracrine signaling, stimulating the neighbors. Or the long distance calls, the endocrine signaling, where signals are secreted into the bloodstream, and that in a completely different organ there is cells that listen to these signals. Right? And then when you look at these signals, you can think about, okay, if they swarm around in this extracellular environment, how can the cell listen to them? Yeah, you need receptors. And so many of these receptors will be present in the membrane of cells. And there's a few flavors of this kind of receptors, like the tyrosine kinase receptors, or the G protein coupled receptors, 7 transmembrane proteins, or the cytokine receptors. But basically the concept is kind of the same. For pharma this is interesting. If you think about where can I have an anchor point to modulate signaling, well, one of the accessible anchor points is these receptors that are present on the cell surface. But then of course signals need to be passed, and so we have these pathways, these combinations of genes that take care of that. This is like quite, well, you have a few of these, but I would say it's not a huge list, and you will see them very commonly affected in different types of cancer, so PI3 kinase pathway, MET kinase pathway, etc. And basically they all end up with signaling to the nucleus, where you tell the DNA, hey, transcribe a few genes, stop transcribing another few genes. That's the consequence of being triggered from outside the cell and how inside the genes will be regulated. Cells can die, sometimes they should die, sometimes they die just because they're damaged, so you really break them. In that case, this often goes by a process called necrosis, so this is kind of blunt damage. But there's also a mechanism called apoptosis, programmed cell death. And really these cells realize, I have to die off. I want to do this in a gentle way in a way that other cells understand this is how it is supposed to be. This is not dangerous, this is normal. And in particular, phagocytes taste the difference between what is abnormal damaged, abnormal cell death versus apoptosis, programmed cell death. So how are these signals induced to die off? Well, this can be intrinsic, withdrawal of growth factors, or really injury, radiation, toxins, free radicals. It can also be by signaling, by receptor ligand interactions. So there's death signals that can trigger cells to die off. Intracellularly, many of these mechanisms come together with caspases. DNA is going to be fragmented. You get these nice blobs, and these nice blobs are then taken up by the vacuum cleaners called phagocytes, like macrophages, and those then basically make sure that things go in a normal, relatively organized way. Something about the cell cycle. So when you have cells that decide that they have to divide, so when there's the signal of proliferation, then a lot of steps need to be taken. And one is that you want to make sure that within the cell the DNA is complete, nicely organized, structured, okay, right, healthy. So there's this checkpoint for DNA damage. And if this checkpoint, if the DNA has been checked and it's okay, then these chromosomes start to duplicate. So now you go to the double amount of DNA. You need to divide this DNA into 2 locations, so preceding the point where you will get from 1 cell to 2 cells. Another checkpoint has to be taken. Is it really well organized? Is this really okay? And if the answer is yes, you can move into mitosis. Now these chromosomes are divided into 2 daughter cells. And then these can differentiate, etc., or at a later point in time decide to proliferate. Yeah. So, what if in this cell cycle, at these checkpoints, the cell actually senses that the DNA is not okay. There's like damage, that there's a chromosome missing, that somehow this is not good. What's then the protection that the cell has? So here we look at the normal cell, and I will focus here on a tumor suppressor gene called p53, or TP53, and the reason is this is the most commonly mutated gene across basically all cancer types, so it's worthwhile to realize why this may be the case. So p53 in a normal cell, when there's DNA damage, it will play a role in this checkpoint for DNA damage. So when there's DNA damage, p53 is activated and it will say something like, okay, stop, either try to repair the DNA, and if the cell succeeds, then you can move on. If you don't succeed to repair the DNA, okay, too bad, but then you go into apoptosis. Yeah? So repair successfully, great proliferation, fail to repair the DNA damage, go into apoptosis. What will happen if you miss p53? What will happen if p53 is not performing the function it's supposed to have? You'll have DNA damage, there's no control, and so these cells will proliferate no matter what. And then you get this great enhanced accumulation of DNA alterations. So where in the beginning we said, well, it's not one DNA mutations, there's many, you can imagine that processes that increase the amount of DNA mistakes that will accumulate, that those processes will really enhance the chances that cells will turn into cancer cells. And this is why in many cancers you will find one way or another defects in the DNA repair mechanisms or in this checkpoint controls. Telomeres. Yeah, telomeres. Who has cultured cells in flasks? And who has cultured primary cells, not like cancer cell lines, but primary cells, fibroblasts? Yeah. Is it easy to culture these primary cells compared to cell lines?
Some yes, some no.
How long could you maintain them in culture? Years?
Beat me, I can't.
No, I don't.
I can't. I will not do that.
Yeah, I only kept them for like a week.
Yeah. Yeah. Why not years?
Well, we were only differentiating precursor cells at the macrophages and then testing the macrophages, so we didn't need to keep them in culture very long.
Yeah. But it's also a challenge to keep them very long in culture, and one of the reasons is for the telomeres, because at every cell doubling, the ends of these chromosomes will not copy completely, so you will start losing these ends of the telomeres. And for cells, this basically means after something like 30 times proliferating, it just becomes too short. If you can repair this, if you can activate telomerase and make sure that these telomeres are lengthened again, we would kind of have eternal youth, as people like to fantasize about telomerase. But yeah, in normal cells, telomerase is not active. So in stem cells it is, and so you can activate it, and this is what cancer cells do to make sure that they can go beyond this proliferation of 30 times, to make sure that the telomeres get sufficient length to go to the next proliferation step. So far I've been discussing processes that matter to the cancer cell, that matter to what happens within a cell. Proliferation, apoptosis, cell cycle, telomerase. But I also, and so basically those are the processes that take place within these cells, which will take place upon signals from outside the cell, right? So growth factors, or what does it say here, anti-growth or death factors. This is signals from outside, cell response. And I showed you some of the things that can go wrong. And the things that go wrong here, if this takes place in cancer, and we see this frequently, we refer to these genes as oncogenes or as tumor suppressor genes. And so one thing that you need to realize is when we look at these oncogenes and suppressor genes, that oncogenes refer to activating mutations. So a mutation in a single allele is sufficient to get this shortcut activated protein and gain proliferative or survival advantage for these cells. In case of a tumor suppressor gene, we are discussing inactivating mutations, and in fact, to really inactivate a gene completely, both alleles of such a gene need to be affected before they will end up in proliferative or survival advantage. So this is just like definitions: oncogene, tumor suppressor gene, how they biologically differ, activating or inactivating. Single allele affected, both alleles affected. It's great fun that it's now possible to do a lot of sequencing. I realize I have some maybe somewhat older slides, but nevertheless, so this is from 2013, and in those days the Cancer Genome Atlas project had been running for a few years, and so people were looking at a pan-cancer effort, thinking about which genes are most commonly affected across cancers, and then you get tables like these, and you can see immediately the most red horizontal line here, so this is different cancer types, is the line where p53 is shown. So this is the most frequently affected gene, and it affects genome integrity. And you can see a number of processes that matter and a number of genes involved. In this paper they came up with 127 genes, 20 categories, blah blah blah. Fine. Sequencing is only becoming more and more available, affordable, doable. And I just want to mention two initiatives that are ongoing. One is PCOG, the pan-cancer analysis of whole genomes. It's kind of the project that follows the Cancer Genome Atlas. And the other one that is also really nice is the sequencing that is done at the Hartwig Medical Foundation. This is next door in Amsterdam. People have heard of the Hartwig? I see a few hands. But it's really in size in terms of, diving into tumor genomes. In this case, this is sequencing of the metastasis. In this case, it's sequencing of the primary tumors. But the numbers of cancers that can be studied here, this is really, according to top of the bill international efforts. So I was discussing these cells and the intracellular processes. When I was young, as a PhD student, everyone was focused on what the hell is happening within these cells. So all research efforts were focused on signal transduction pathways, discovering p53, understanding what the function of p53 is. But at a certain point in time, when people had a few clues about signal transduction, they started to realize, okay, cancer is not an empty bag with a few neoplastic cells. Cancer is neoplastic cells in a tumor microenvironment. This switch came, around 2000, not before. Before that, people were really only focused on these cancer pathways. But this is kind of the view how you, can imagine that cancer cells in practice live, interact with other cells, with non-cancer cells. And so there's lots to say about tumor stroma, where you can see that, here, if you here have the epithelium, in case of colon cancer, these would be the cells that turn into cancer cells. You have here the basement membrane, you have here a loose matrix, and swimming in here somehow are fibroblasts and immune cells. And you have the wiring called the vasculature, the blood vessels or the lymph vessels. And basically you have all these interactions between cells, in this autocrine, paracrine, or endocrine signaling way. So there's lots of communication going on. When you then think about this blob of cells. So these cells start to proliferate and they become like a big blob and a bigger blob. What are the problems that this blob of cells is going to encounter? Will it be able to get oxygen? At a certain point in time, not anymore, because if the blood vessels are not close enough and you get the diffusion of the oxygen from the blood vessels to these cells, so these cells are kind of suffocating. And cells don't like that, so we have genes like HIF1 alpha and oxygen-inducible factor 1 that will make sure that if the cell starts to suffocate, that there's going to be a glycolytic switch, that there's going to be production of a protein called vascular endothelial growth factor, which stimulates angiogenesis. So basically it says, I'm changing my metabolism. Okay, it takes more glucose. And okay, with a few more doublings, also the glucose diffusion limit is going to be a problem. But anyway, for the time being, switching to glycolysis will help. And inviting blood vessels to come over will help. And then, it's further proliferation and then new trouble ahead, but at the same time with a more acidic environment breaking through the basement membrane, being able to get into the blood vessels, and oops, problems increase, at least for patients. So here you see endothelial cell layer, cells loosening up, having to travel through this basement membrane, and then when breaking through, these cells will, with their interactions of inviting the blood vessels to come over, they may end up in the blood vessels, they may travel, and at some distant location, they may form a metastasis. Make sense? I find this a hell of a complex thing to do for a cell. So first you are like connected, making friends with your neighbors. Then you need to travel on your own. You need to survive that. You need to be strong like a stem cell, which is why you get sometimes these stories about cancer stem cells. And finally, when you're then completely independent, you need to settle again in a new environment. So really, this seems like a sensible picture, but I think biologically this is extremely complex.
By far most tissues are not, the cells and tissues are not mobile. How do these cells become mobile so that they can travel through another layer of cells and get into the bloodstream? That seems to me.
I think many cells are very mobile.
Yeah?
Yeah. It's really nice. You have now these microscopes, and you can label cells with fluorescent dyes and put a microscope on top of it and make beautiful movies. And it's amazing, I now start to think of us as a blob of continuously moving cells. It's fantastic. Try to look up some of the movies on how people are now capable of filming cells. It's really dynamic. It's, the immune system. One other trick that cancer cells need to do is to escape from the immune system, right? We have an immune system, and so when there is strange bacteria entering our body or strange viruses, our immune system, most of the cases, is doing a great job, right? Protects us. Problem of cancer is, of course, what is really changing? It's kind of a cell from a person changes a tiny bit in a few genes, maybe in a few proteins. Can this truly trigger the immune system to recognize this as danger, as something that should be deleted? But apparently it can in a number of cases, and so cancer cells have thought about how to avoid being attacked, being recognized by the immune system. So in a normal situation you have a cell, and in those cells there is proteins that are being chopped up, and then they're presented, like a waiter presenting on a plate a few beers or a glass of wine. And so these T cells can recognize this, And these T cells have this huge repertoire of sensors to think like, okay, this is something that I have seen before, or this is something that is really strange, and now I know whether I have to react aggressively or friendly. What if you don't present the antigens? Well, if you don't present anything, the T cells cannot recognize it. What if you delete the plate that the waiter uses to present it, right? You cannot present any more these antigens, so the immune system cannot recognize it. Or what if you have this nice presentation together with a story like, shut up, don't be activated. You see these little yellow balls, the signals that kind of say, be quiet, don't respond. So, when I did my PhD studies in the 90s, there was this huge hype about immunotherapy. And many immunologists were fantasizing all these wonderful tricks, how they would cure cancer. And really, kind of nothing was successful. Lots of technologies, lots of cell culturing, lots of understanding of the immune system, lots of knowledge. Basically nothing worked. Has this changed recently? Have you heard about immunotherapy? If you now go to a cancer meeting, you cannot avoid the word immunotherapy. There's immunotherapy trials everywhere. So the Nobel Prize was given to the scientists who came up with the so-called checkpoint inhibitors, antibodies that block CTLA-4, PD-1, PD-L1. And you cannot go anywhere anymore where people will say, oh, we need to do immunotherapy. And I understand the enthusiasm, because I think when you look at the stories, there have been some amazing, amazing recoveries of cancer patients by immunotherapy. At the same time, I think we are now at a point where we still do not really understand, out of 100 patients, which 10, 15, 20 will respond really well to immunotherapy, which 50, 60 are not responding at all. So we need biomarkers to think about, who should be offered immunotherapy, who not. And basically this goes for any cancer medicine. But anyway, this has been a very exciting development in recent years, and in the clinic this has become I think a big game changer. So I guess I went through all of these biological processes. I'm running a bit out of time, that's okay. So as mentioned before, per biological processes, a lot of genes are involved. Each of these genes can be affected in various ways. And I will go very quickly, because I think many of this you kind of know. So you can damage DNA, and this can happen in many different ways. And for each of these DNA alterations, there's some kind of DNA repair mechanism that is present. So there's different machinery to repair different kinds of DNA mistakes. And in a number of different cancers, sometimes you will see that different subtypes of these DNA repair machineries are broken. So it matters a lot whether in a tumor mismatch repair is deficient, or whether p53 controlling chromosomal gains and losses is mutated. Right? DNA mutations, as mentioned in the beginning, if you alter DNA and you do this in a position of a gene that matters functionally at the amino acid level, then this DNA alteration will affect RNA, will affect the protein. Then there's many ways in which you can also alter the DNA. So there is, like within chromosomes, there can be chromosomal breaks, there can be translocations, there can be fusions, there can be deletions, there can be amplifications, regions that are highly amplified, And so if you would look them up in a cell by immunofluorescence, you get a huge crowd of these orange speckles, basically indicating that certain genes have been amplified many-fold. There can be modifications at the DNA level, like methylation of the cytosines, but also methylation, acetylation of histone proteins, which affect the way that these regions in the genome are being transcribed. Quite often we would say that methylated DNA usually is related to lower expressed genes, although there's always exceptions. And likewise, alterations at these histone proteins matter to the ease at which genes can or cannot be transcribed. And there's post-transcriptional modification. I'd like to give the example of microRNAs. You know, for many years I never heard of microRNAs. One day there's microRNAs. Okay, what do? Well, you have these RNAs, and are they kind of stable? Will they survive for a long time? Allow many proteins to be produced? Well, that depends a little bit. If you have microRNAs and they determine whether these messenger RNAs are stable or unstable, they will also determine whether you will have a long or a short time in which certain proteins can be produced. So that matters to the transcriptome, or actually to the protein composition of cells. So a layer of regulation of the messenger RNAs. And then we have germline variation. I don't see identical twins here. And so, you know, we are all different. How different are we? Maybe 1 in 1,000 nucleotides. And then you could say, we are 99.9% alike. I would say, well, most genes have more than 1,000 nucleotides, so I guess we differ in all of our genes. And it's more than sufficient to be all unique. But of course, the way that we are programmed in our germline matters to the kind of somatic alterations that are going to be more or less effective to turn cells into cancer cells. Right? And this is the most complex one of all. Computational, I think the hardest one to deal with, which is why I think either we get to boring research studies or to non-relevant ones. But keep in mind, Mary was great in tackling this problem. But I really think this is one of the most complex problems that people don't dare to tackle because computationally, statistically, we're still not there. Alright, getting back to complexity. So we have all these biological processes, many genes involved, many ways they can be affected. And if we put this all together, you know, there was these different flavors of genome instability that kind of enhanced all these changes that took place. And so in case of colorectal cancer, this is like microsatellite instability, chromosomal instability. I did not talk about that yet a lot. And there's, you know, etiology, so environmental factors, genetics, genetics in terms of sporadic disease or hereditary factors that play a role, environment, diet, what do you eat, do you practice a lot, exercise. And all of this builds up to this phenotype basically being this tumor that on the outside, you know, one colon tumor on the outside looks kind of the same as the next colon tumor. So if I ask you the question, if you look at this phenotype, if you look at this outside of this colon cancer, do you now have a little bit of a better understanding what we mean with the complexity of tumor biology? So the things that are underlying. I see a few people nodding, a few people sleeping, I guess. Alright, yeah, you know, pharma. Pharma has done for many years, they've practiced wishful medicine. For many years they were thinking like, we should find this one pill that cures all. And I must say, I think this one pill already has been invented a long time ago. I think really aspirin gets close. But it doesn't solve all the problems. So, you know, this basically doesn't exist. and as you now hopefully understand, looking at the diversity between patients, between tumors, we can easily understand that cancer patients need to be subdivided in a way that we can actually understand which patients do not benefit from a drug while the drug is being toxic to the patient, or when the drug is beneficial, also toxic, but okay, or not toxic, not beneficial, not toxic, beneficial, but we need to be able for each drug basically to understand who responds well, who doesn't. Molecular profiling, I think, is key to get a better understanding of tumor biology, which tumors do not respond, prognosis for the patients, who is likely to have a tumor that is cured when the surgeon takes away the tumor, who is likely to develop a recurrence and should have adjuvant chemotherapy following surgery. And for all of this, really, molecular profiling is badly needed. And to do that, technologically, yes, DNA sequencing, RNA sequencing, mass spectrometry are great technologies, but we also need people who know how to deal with these data. So I'm glad you choose to study bioinformatics. When you look at this wheel of Hanahan and Weinberg, by now, well, this is still 2011, so the examples only increase, but basically for each of these biological processes, people have been thinking about drugs that may be beneficial to the patients. And I think that's about it. Time for a coffee break, and then I will move on to examples for. Alright, let's continue. Maybe the first question is, were there any remaining questions on the lecture of the previous hour?
Yes. No.
No burning questions? Okay. So, a little bit more practically. I'm working in a group, we name ourselves the Translational Gastrointestinal Oncology Group. And what is it that we do? Well, from large cohorts of patients, we collect clinical information. And from colorectal cancer patients, we try to collect tumor tissue, tissue of the normal colon, blood samples, stool. You may be familiar with the population-wide screening program in the Netherlands. We try to improve that, so we have the most expensive collection of shit of Europe. We have a model organism, a model to study tumor biology, that is organoids. So organoids, human organoids from normal colon, colon adenomas and cancers. And what we like to do is to do DNA, RNA, protein profiling. What is kind of missing, one could argue, is profiling of metabolites. Of course, this is something that is important, but it's not something that we at present or in the past really have been doing. So, a summary like this you may find from many different labs. But the other thing that we do is actually to really take an effort to move from molecular profiling of tumors to translation of molecular knowledge into clinical tests. And this is an easy sentence, but there's actually a lot of work behind this. So we really invest a lot, well, as I kind of mentioned, in building biobanks, but also in improving research IT infrastructure, which is actually one of the reasons why in the past I got connected to Sanne Abel to really run some of these projects, and in addition to the scientific projects. So, as I kind of already emphasized, modern research is really increasingly dependent on bioinformatics analysis of molecular profiling data, and there's all kinds of data that can be generated, all kinds of data that need to be analyzed and need to be integrated. What about colorectal cancer? Well, if you ever present your work, the first slide also always shows how incredibly important the problem is that you try to solve. In case of cancer, this is always an easy case. Many people die of cancer. In case of colorectal cancer, many people actually get colorectal cancer. But there's also like a message like, where should we focus? Well, early detection, stage 1 disease, has a huge 5-year survival rate, whereas when diagnosed in the late stage of disease, metastatic disease, there's a less than 10% 5-year survival. So this is one of the reasons why in the Netherlands and in many European countries there is this population-wide screening program, and you may have seen in the news not too long ago that actually this program works. Really, by now, more people are diagnosed with colorectal cancer in its early curable stages. There's really a stage shift towards stage 1 and stage 2 disease, away from stage 4 and stage 3 disease. So this is why truly the Dutch program, or any of these screening programs on colorectal cancer, work. Here's the sequence of these normal to cancer as the pathologist looks at it. What do I want to show in this slide? Well, we already discussed all kinds of genes that are activated in the transition from normal to cancer, kicking in of genome instability, in case of colorectal cancer, microsatellite unstable, chromosomal unstable. And what we try to do is to come up with biomarkers. Biomarkers for better diagnosis, prognosis, or prediction. Prognosis meaning who is cured by surgery alone versus at risk of developing a recurrence, and predictive, what drug matches which particular patient. I adapted the slides a little bit compared to previous years, so I have to remember what I tried to say here. I think we're moving to an example of prognosis. And so for prognostic biomarkers, one could argue if you have stage 2 or maybe stage 3 disease, the surgeon takes away the tumor, how likely will the tumor recur? And if that's likely, you would like to offer patients additional treatment like adjuvant chemotherapy. If it's unlikely to recur, you don't want to offer the chemotherapy because it gives a lot of side effects and it's really a burden to the patient to undergo these therapies. Now, I made this case of cancer is a disease of the DNA, and so I'm terribly frustrated that I have not clear DNA alteration overviews that come up with great prognosis. People have been a bit more successful by looking at RNA expression profiles. And one way that my colleagues would explain to me why they are kind of successful is, well, you know, if you look at the RNA, you have some kind of summary of what's ongoing in all these DNA, and maybe the summary is kind of informative enough to come up with some prognostic values. So at a certain point in time, people started to do RNA profiling, and the people who did this first were the people working in the field of breast cancer, and one, I would say, still famous example from the Netherlands, from Netherlands Cancer Institute, Laura van 't Veer, who's now actually at UCSF, is the development of the mama print. Who has heard of the mama print? Recently made. Also, mama print has been in the news not too long ago, whether by now finally this should be funded by the Dutch health insurance or not, etc. So anyway, people started to do these RNA expression profiles. Mama print is one example, Oncotype DX is another one, and basically you do this profiling and you decide likely to get a recurrence, unlikely to get a recurrence. Probably wise to provide adjuvant chemotherapy, probably wise not to give chemo. So, quite a number of years later, a number of groups, roughly at the same time, started to do RNA profiling on colorectal cancers. And so one of the groups at the AMC, Jan-Paul Medema, Louis Vermeulen, came up with this paper on classification of tumors in 3 categories. They named them CCS 1, 2, 3. And what you can see on the what you can see here is roughly these 3 groups. And what you can see here is the survival curves where there's this one subtype that has a worse prognosis than the other 2. And so basically there were 6 papers at a time. You don't summarize them here further. I could have done it. By now what I should have added is these groups sat together and they actually decided, let's come up with a consensus molecular classification, CMS. I'll put it in the lecture next year, Bas. The CMS classification is CMS 1, 2, 3, 4, where also there is this one subgroup, in this case CMS 4, that has this worse prognosis than the other 3. So somehow we have been trying on the RNA profiling, can we distinct these tumors in this way? Yes, to some extent we succeed. Is it really, really, really helpful? This is something that I think many people are finding out at the moment, trying to think about how to apply CMS classification in practice on clinical samples. And I think there's lots of efforts still needed before this is going to be realized. So as I mentioned, I was kind of frustrated because, theoretically I would argue it should be in the DNA. DNA is important. Why can't we just look at DNA alterations and come up with good signatures, etc. So when you think about all these DNA alterations, then one can think about different steps. One of the PhD students that worked on this is actually Evert van den Broek. He's now becoming a pathologist in Groningen, and he has been working on this. And so, if you look at DNA alterations, what we know is the point mutations that have been categorized reasonably well. And this is an example of the Cancer Genome Atlas Network, paper from 2012. Hundreds of tumors were sequenced, and then in case of colorectal cancer, p53 frequently mutated, 60% of tumors. This is all chromosomal unstable tumors, I must say. APC in even more often, but this is quite specific for colorectal, that APC tumor suppressor gene is frequently mutated. So the frequency of APC will be much lower in other tumor types, but in colorectal it's reasonably high. Now if you move further, you can see here KRAS, 40%, and then TTN. This is probably a sequencing artifact, because I never see TTN, titin, never hear any oncogenic activity there, so it's nice in this list. Probably doesn't belong here. And then it drops quickly, PI3 kinase, etc., etc. And here we are at frequencies of genes that are affected in 3% of the tumors. And then this tail of genes that are mutated at low frequencies is endless. Yeah? So there's a few genes mutated at high frequency, many, many, many genes that occasionally get affected. And you can imagine that if you want to study a gene that is affected in 40% of cancer cases, hey, if you have 100 cancers, that's nice, isn't it? 40 with a mutation, 60 without. Sounds like reasonable numbers. But if it's mutated in 1% of cancers, how many cancers should we have sequenced to study these kind of genes? And I think this is the kind of transition that we are now moving in, that the numbers are really increasing into the thousands, tens of thousands, hundreds of thousands, going to the millions. You'll be the people analyzing the millions of whole genome sequences to get answers to these so-called, low frequency mutated genes. What is nice about these kind of mutations, they were identified. Remember the first plot I showed, which included APC and p53 and KRAS? So even in this first old Vogelgram with these steps of genes being affected, those are actually the most prevalent mutations. And for these genes, a lot of functional studies have been done, so we have a reasonable understanding how these genes affect cells. When you look at chromosomes of a cancer cell, it's different from looking at chromosomes at a normal cell. In a normal cell, for each chromosome we have 2 copies, right? And in a cancer cell this can be a mess. And in colorectal cancer, 85% of those suffers chromosomal instability. And this is something you can see here. So here you have the chromosomes that are painted, each chromosome in a different color. But you can see that the numbers are not always nicely 2 of a kind, right? There's a lot more over here. And sometimes you get these odd chromosomes where you have multiple colors. So apparently there's copy number gains, there's copy number losses, numerical changes, and there are structural changes where you see multiple chromosomes glued to each other, translocations. And so here I think we have far less understanding. Now what in our group we have been doing a lot is looking at these DNA copy number aberrations. So in this case using array CGH, so each dot is like a probe on these arrays, and when it's more frequently present it goes above the 0 line, and when it's less frequently present below. And so if you go from chromosome 1 to 22, you can see where there's gains and where there's losses, right? And this has been done with array CGH, comparative genomic hybridization. By now you would do this by whole genome, but shallow sequencing, so just 0.3x is sufficient to generate these kind of plots. Now when you look at these profiles for hundreds of colorectal cancers, you will conclude that this is not necessarily a random profile. A lot of events may seem to be random, but there's also a lot of events that will recur. 20q gain, 13q gain, 8q gain, 18q loss, 17p loss. This is something that is very typical to happen in a colorectal cancer, on top of whatever other alterations that one can see. But that doesn't mean that we understand when there's 20q gain what really drives 20q gain. Was that one gene on 20q? Was that many genes on 20q? What really is going on? So there's a lot of knowledge that we're still lacking. And what we have been neglecting, I would say, is in the field of cancers to look at structural variants, to actually focus on chromosomal breaks. And you can imagine that for lots of reasons, there's really breaks in chromosomes by small deletions, insertions, inversions, interchromosomal translocations, or interchromosomal translocations. And by now one can make these beautiful circle plots and indicate where all the trouble is. But it's interesting. If you think about leukemias, the first thing molecular biologists think about is translocations. BCR-ABL, fusing one gene to another as a key driving event. And in the field of leukemias, they kind of neglected the chromosomal gains and losses for many years. And for the people working on the neoplastic cells from epithelial origin, like colorectal cancer, for many years only focusing on copy number gains and losses, not thinking about these translocations. But they are there, they're abundant, and they require further investigation. And you can see them easily. I mean, look at here, this is a small piece of a chromosome where you can see losses, gains, and you can see all these jumps. And basically you can just take your pencil and draw a few lines, and you understand that somewhere between this probe and this probe, the chromosome must have been broken, right? And so Evert van den Broek was kind of looking at a large series of colorectal cancers where we had these array CGH profiles, and these series were actually derived from Dutch trials, the Cairo and the Cairo 2 trial. And so these trials in itself are already quite famous. So for medical oncologists, this was really informative to what therapies patients respond or not. These are all patients with metastatic colorectal cancer, and so of these 350 we had these copy number profiles. And on top of that we looked at point mutations, at least in the most frequently occurring point mutations like APC, p53, KRAS. And with that we tried to combine information and think about, can we do something that people did for this RNA profiling, coming up with these clustering plots and seeing whether there's groups of patients that do better or worse, etc. So here you see a summary plot on chromosome 13 in this case, where we looked at this breakpoint position, and this is the percentage of the 350 tumors that were affected, and if you see this large spike, it means that somewhere there were multiple tumors being affected at that position, and when you see nothing, well, not affected, or the single tick just in a single tumor. And then Evert started to overlay these positions with genes, asking the question, well, if the gene has a certain size, and I see frequent aberrations in there, how often are these genes affected? Can we do some statistics? Is this more than random, less than random? And actually a lot of genes came up that are likely to be affected in a non-random way more frequently by these chromosomal breaks. And when you try to plot this for this group of tumors, for this cohort, again, p53, APC, KRAS in this group of tumors, the most frequently mutated, but then actually other alterations came up affected by these chromosomal breaks, genes like MACRD2, PARC2, FHIT, etc. So looking at it like this was like, oh, that's funny. So we have been focusing on these point mutations, which really matter to a large number of genes. But if we now add on top of this, thinking about genes that may be disrupted by the position of chromosomal breaks, there's going to be another huge series of genes that really may matter to these tumors. Of course, one of the key questions is, if you see all these breaks, are we looking at an epiphenomenon associated with a DNA repair problem, associated with chromosomal instability, or are we looking at something that is clinically and biologically meaningful? And so, together with Sanne, with Maurits Dijkstra, we applied a tool called network-based stratification, which tries to make use of gene knowledge to classify patients. So you try to group together genes that functionally may do something together. Remember the pictures I showed you of these cancer pathways where people try to group everything related to apoptosis, everything related to angiogenesis. You will be able to think of smarter ways to even in a more functionally relevant way group these genes together. And then you could argue if in such a functional unit one or the other gene is affected, doesn't matter whether it's one or the other, but the unit is affected, then we can build up from these modules and see how we can classify these patients into subgroups based on the kind of functional units that are affected rather than single genes that are affected. So this is what we tried to do, and then we came up with a plot like this. And so, you know, it's always nice to come up with something that you can say, oh, we see 4 subgroups. One of the things I learned is that you need to think carefully about, why don't I see 3 subgroups or 5 subgroups? So there's all kinds of choices that need to be made, and some of them, you know, are a bit arbitrary, but you have to think about the most logical ways to make such decisions. And then you can look at survival curves, and what you should know is colorectal cancer patients with metastasis, they really don't survive well. That's what I showed you in the beginning, right? Less than 10% 5-year survival rate. So seeing one survival curve that is even worse than the others is kind of exceptional. And if you pull the other three together, just to make the figure more clear, it's really surprising to see that indeed there is this one group that really performs really bad. I had not expected that in this patient population, trying to make this subclassification of patients, that we would be able to come up with a difference that clinically means a 7-month difference in median survival. So then people said, well, you know, Raymond, you are so enthusiastic about macro-D2. Did you talk about macro-D2 at the BioSB?
Yeah.
Were more people at the BioSB? Did you hear Nienke's story? So you hear already the story. I'm enthusiastic about macro-D2 because, you know, apparently it's very frequently affected. But it's also a problematic gene, and the first problem is it's big. It's probably the biggest gene, around 2 megabases or so. So, you know, chances are that at random, if you have a lot of breaks, macro-D2 is going to be broken. And macro-D2 is also considered boring because it's probably a fragile site. What's a fragile site? It's something like, you know, you start to proliferate, you divide, and there are these regions in the genome that are the first to split up the DNA, duplicate, and there are regions that are the last. And so, you know, if these are just the weak spots in the genome, so be it. And so some people challenged me and said, well, you know, think about microsatellite instability. What's the problem with microsatellite instability? It's DNA repair of these dinucleotides, these repeats. So if you have 1 out of 4 genes mutated, MLH1, MSH2, MSH6, BMS2. If 1 of 4 is mutated, everywhere in the genome you'll find changes. So what if you have a general problem here? And this general problem is caused by gene X, and all your fragile sites are going to show these alterations. So I was very happy that last year a few papers came out that actually said macro-D2 functionally really matters. And functionally it relates to DNA break repair, DNA double-strand break repair, in a way that macro-D2 has a role that is connected to PARP. These little groups are poly-ADP ribosylated groups on a certain protein called PARP, which is needed for double-strand break repair, and then macro-D2 is needed to take off the last bit of these ribosyl groups. And then this gene, PARP1, can function over and over and do excellent double-strand break repair. If macro-D2 is disrupted, this process is gone. So, one message is, you can be enthusiastic about genes that are affected. Biologically, you need to come up with explanations how this may work. At least these presentations, these publications gave some clues why actually the gene macro-D2 may be of interest. Evert van der Broek did a number of these studies. Roel Glas has done some work on this, and more recently Nienke did. And Nienke had to step in at the BioSB, and I know Nienke did both a great job in her internship and is very well capable of presenting. Maybe next year, Nienke, you have to give the second part of this presentation. But congratulations, because you won the prize for the best presentation as a Master Bioinformatics student. Yeah. I think that deserves an applause. Nienke has been playing with data from the Hartwig Medical Foundation. You were number how many to get access to these data?
Number 16.
Number 16. You feel like a front runner, right?
Yeah,
You are.
We don't know what the other people did.
So, Hartwig Medical Foundation does whole genome sequencing. And it does so in a way that for Illumina sequencing it's like 100x deep on the tumor part and 30x on the normal DNA. So you have matched normal with each of the cancer tissues. This has been done now for something like 450 colorectal cancers, but this really goes into the thousands because it's not restricted to colorectal. Hartwig has data on any type of cancer where metastatic lesions could be harvested for whole genome sequencing. So one of the questions that Nienke addressed is, how does this actually look like? Does this kind of relate to what we have seen by RACGH? So Nienke made beautiful plots to come up with overviews on what kind of alterations can be seen here, basically confirming that structural variants are highly prevalent. You have to imagine that when we started off looking at structural variants based on sequencing data, there were bioinformatics colleagues who showed me 5 different tools that could do the job. And they like to compare the 5 different tools, and sometimes you have these analyses and then it's nicely overlapping, right? A few percent off whether you use tool A or tool B. But these were the kind of tools where 98% was non-overlapping, where you wonder, okay, what is the strength, what is the weakness of each of the tools? Sometimes you have all the right hits and 1,000-fold rubbish on top of it. Sometimes you have all of your hits, most of them have the right structural variants, but basically you missed out the far majority that is present. So this is the kind of challenges that you're all tackling or going to tackle, thinking about data analysis, etc. And I think for structural variants this has been difficult, and so we were kind of skeptic that the pipelines being used by the Hartwig would actually really yield a number of hits that might work. But anyway, I think the Hartwig has done a reasonably good job. There was quite some concurrence between the data as they had generated, something we could recognize on our work, and on top of that still a lot that needs to be validated anyway. Lots of Nienke to show that indeed MACRD2 also in these data sets is one of the most prevalently affected genes. This is on the sample level. Many of these samples actually had multiple breakpoints per sample. And coming up with plots that I like. And this is about the visualization. I'm really happy with bioinformaticians who like to think about how can I show everything that I know to my colleagues that are not bioinformaticians, that are either molecular biologists or medical oncologists, but how can you convincingly show what the point is that you want to make? And of course there's lots of tools around, like IGV, the integrated genome viewer, but then think about how to put in data in these tracks, and maybe you have all kinds of projects where you creatively can think of new visualization methods. Think about one day there was nothing like single cell sequencing. One day there is. How do you show this to people? And besides Illumina sequencing, we are moving to PacBio sequencing, long read sequencing. If you do long read sequencing, I hope for bioinformaticians it's easier to really see how all of these breakpoints are truly put together. Because if you have these chromosomal breaks, it does not necessarily mean that you have the end of one part and then the end of another part, another chromosome glued together. No, quite often there can be some strange nucleotides intermingled, and this makes the job to recognize which pieces of genome were glued together will make it more difficult. Or you're looking into these repetitive regions for which it's very hard. So this is another approach that we take to tackle our investigations for structural variants. And this is, I think, the approaches that we are taking at the moment. So you have DNA alterations, point mutations, copy number aberrations, structural variants, where genes like MacroD2 are highly prevalent, but there's a long list of genes relevant here, moving to long-read sequencing. And one of the things that the whole class has been working on and that we want to pursue is to think about ways like, if you have so many candidates, can you come up with machine learning approaches that give a clue like these candidates are likely to have more impact biologically, clinically than others? Because if you think about characterization of a gene functionally, like the example of the MacroD2, these functional studies, the wet lab work is really slow, really tedious, laborious. It's great fun, but it really takes years to understand some of the functions of a single gene. So in the past, I think for molecular biologists, it was like waiting for bioinformaticians to catch up. By now, I think many bioinformaticians need to team up with wet lab people to think about how can we, in a productive way, come up with confirmation in the wet lab, what actually comes out of the bioinformatics analysis that can be done. I think this is about it, what I wanted to say. Summary. Many genes with recurrent breakpoints have been identified, so this is a category of genes that I think is worthwhile to pursue how they may affect human biology. They can function as CRC drivers, complement oncogene tumor suppressor gene mutations, but really a lot of studies still need to be done. I think I convinced you, right? That if you enjoy the study that you're doing, that at least you've made a great choice in terms of perspective for the coming years, all exciting research coming up, and all kinds of companies that want to turn this into great applications. So I think it's really a great and dynamic field to be in today. And I'm very happy to have seen so many of you do internships with us, often in collaboration with Sanne Adon or with Bas. Apologies to some of you who have not replied your mail yet, but looking forward to new candidates to come. Thank you for your attention. Questions? Are there any questions?
Alright, so no questions is either a very good or a very bad sign.
Maybe.
I do have one question, but it's not about the biology. But it is, how would a normal person that doesn't have cancer check if he has cancer at some point? Do you need to palpate yourself? Do you need to get screened sometimes? What's your advice on this?
So there will be different answers to different types of cancer. I think what so there's no straightforward, clear-cut answer. I think what is nice about a country like the Netherlands is that we have health insurance for everyone, and that means that there's in fact a drive, a stimulus to try to prevent people from getting cancer in the later stage of disease. So, you know, joining the population-wide screening programs would be one approach to try to detect cancer as early as possible. And I think there will be other ways in the future in which this can be done. One of the studies that I'm working on that may not be excellent for early diagnosis, but who knows, is looking at free circulating tumor DNA. So we have great collections of blood samples where we try to look for DNA mutations there. But this is a field which is like immunotherapy. It's a hype, which is to some extent, you know, don't be too enthusiastic too soon. Maybe the great applications come in 20 years, like what happened for immunotherapy. But there is a lot of momentum. There is a lot of thinking about how to read, understand cell-free DNA in the blood. And so there is lots of potential there in many applications.
continue on that. This is not something that you do yourself. Right? This is something that you go into a clinic for, or that you get referred to by a general practitioner. So, like the question started with palpating yourself. This is, well, for some tumors that might work. For others, you do need an expert to step in.
Always go to a doctor when you feel uncomfortable and are in doubt. But I hope that was clear to everyone. But as you know,
Or just figure symptoms.
So that
You're guaranteed.